512. After this, the next version is gradient. So here is the option of 5 flow cells you can keep together. 72 hours is the runtime. Up to 200 GB data you will get from this theoretically, but in actual practical up to 150 GB, and available channels up to 2560. The latest one is ProMethines, up to 48 flow cells you can run in this runtime, 64 hours, up to 15 TB data you get, but in actual 8.6 TB, and up to 144,000 available channels are there. So whatever the maximum yield and theoretical yield is written here, it is the sum of 48 flow cell. So if you calculate for one flow cell, so it will come around 150 GB in case of promethane. They have also P2 solo device which has the option of keeping only two flow cells. So in that case, there is the yield which we got is around 150 GB each flow cell in promethane case. So this is the real-time monitoring through the Mino software after sequencing. So this is the actual photo which can be performed earlier, few months back. So you can see here the flow cell ID, sample ID, available pores, those nanopores, how much they are present, runtime, active status reads, estimated basis, base called basis, every information you can got. Active pores which are doing sequencing, pores available, unavailable, everything should be present there. You can see pore occupancy here, 88%. You can also see the software, the read length histogram. What is the read length of your current library which are you are running in this promethane, Mina, and Gradine, whatever platform you are using of nanopore sequencing. So after this, NGS is just a technology to generate data. Whatever technology we discussed till that first generation Sanger sequencing, second is that Illumina, third, then PacBio Smarty, fourth Oxford nanopore sequencing. So these all technologies are for just for generating the data from your sample by preparing the library. Now the scientists need to essay in order to get the questions to answer what they want to do with that sequence reads. The variety of problem, scientific field, target, molecule, biological mechanism, whatever is your desired question to answer, you can analyze their data according to that. So there is a general scheme how we will process this data. This is mostly done by dial-up peoples. Uh, so QA, QC, the first step, high error rate, individual cross platform. Then the mapping of these dates has been done, like reference or de novo, and sequence alignment. After this, variant detection means variant calling, and artifacts such as heterogeneity we will see in variant detection. Further annotation has been done, external knowledge, gene function unit. Then further, the last step is modeling interacting networks. So people call these days AIML. So different model you can see like this. So this is the schema how these things have been performed one by one. So in sequence analysis, we generally got FASTQ file. Then we do the read cleaning. The clean reads are in form of FASTQ. Then we do the assembly with the reference or de novo. Then annotation, GFF, or we can directly map and get BAM file. So what happens this raw BAM file? In here, what we will do, we remove the duplicates, realign the quality recalibration, and proceed with the SNP calling BCF or differential expression through this BAM file. So like this way, the data acquisition was done, then the analysis has been performed in case of dialog. So there are different sequencing one by one steps to analyze data. So first, primary base calling has been done. In Ilmina, we will do after sequencing, but in case of Nanopore, you can do even in real time. The signal analysis base calling, we get quality score raw. Then in secondary analysis, we map the reads, quality score B, C. Then the variant calling has been done. Parent analysis, haplotype inference, gene depth analysis, then visualization. And in tertiary analysis, depends upon your what question you want to ask through this the generated data. On the basis of that, you will use that in your diagnostic and therapeutics. So primary analysis, there is base calling, means whatever signals you are getting, to change them into ATGC, the readable format is called base calling. In case of Illumina, it is done by some softwares after sequencing. But in case of Nanopore, even you can do real time, or if you are interested later, then you can do later as well. Then the trimming is the second step. Removing the parts of reads, means whatever reads you got, from that you want to remove some parts of read like adapters and barcodes. Barcodes are for multiplexing your samples earlier at time of preparation library and sequencing them, and later now you are going to analyze. So on the basis of these barcodes, you can identify the particular sample and you can identify them separately. Remove the part of read that is low quality. Some reads you can see have means low quality than below our quality score, so we can trim those read as well. So this is quality control has been done, and we got fast QC, the help of fast QC or semester tools. Then base quality value has been valued in form of Fred score. What is the Fred score? So Fred score means the Q is minus 10 Q power log 10 P, and P is here the Fred score. So let's take the example. If Fred score quality is 10, so it means the probability of incorrect base call is 1 in 10. So base call accuracy is around 90%. If the Fred quality score is 20, then probability of incorrect base 1 in 100. So the base call accuracy around 99%. If Fred quality score is 30, then probability of incorrect base is around 1 in 1000, or the base call accuracy around 99.9%. Like this way, 40, 50, as much you increase the Fred quality score, the base call accuracy will also increase in the same manner. In secondary analysis, the alignment of DNA reads has been done with the help of reference, Then the alignment and mapping of these DNA reads has been done. And then the variant calling has been done. Means now we will use statics here. Means we got different means peaks which are one upon another in this chromatogram. So how you will decide? Suppose in here is one example. You can get read for two bases one upon another. So which has the maximum peak? We will go with the statics. So we will consider the peak which is uh, more height than the another peak. So we will consider that this is the real base present there. So with the help of statics, we will and depend upon the amount of coverage, the data we got from the sequencing. On the basis of that, we will do the variant calling and we decide which base is at which position. We got multiple peaks in the chromatogram. So for that, the maximum coverage, we will rely more on that. So after that, the annotation has been done. It is a in annotation, you will see each thing, which barcode you ligated, your sample name, in which chromosome position, reference variant, gene ID, then genotype, frequency, quality, coverage, mutation, silent mutation, intron, polymorphism, whatever is there, at which the side of mutation, everything you will got after annotation. So again, the variant calling has been done on the basis of this variant calling. We will get the SNP, MNPs, or indels means deletion is there in between reads. So we will see this in the variant calling process of data filtering. So this is all for overview of next generation sequencing. If you have any doubts, so then please ask. Hello. After this, we will discuss how we'll do the hands-on wet lab sequencing protocol. So before that, if you have any doubt in this, so you can ask. Hello.
Sir, will you be sharing this PPT with us?
Hello. Yes.
Sir, will you be sharing this PPT with us and would we do the hands-on today only?
After this, we will discuss that presentation as well. So this is just overview of next generation sequencing from first to fourth generation. So if any one of you have any doubt, because there are so many slides, so I presented them very fast. So if at any slide, if you have any doubt how this thing has been done, so we will discuss that first, then we will proceed with the next PPT.
Sir, I have a question.
Huh.
Sir, yesterday we were studying base pairs. We were seeing the base pairs and they were assigned some quality values. And they were named like I, J, and H. So, sir, can you please explain the quality values of I, J, and H?
I, J, and H. Okay. At time of data filtering, na, in last slides. So IGH. I have no that much idea of IGH. The Preeti will present after me. I think she will discuss about that dry lab person because I'm the wet lab person. So what is the IGH? She will explain better.
Okay, sir.
Any other question?
So, can you please explain the variance type, the part you're asking?
Hmm?
Variance,
Hello.
variance. Hello.
What you are asking? In which slide? You remind me the slide number?
So the